talk today. I was telling Kermit before we started that a lot of this is mostly going to focus on sort of what I'm doing now. Uh, it's not going to have a whole lot of results in the sense that, and the reason for that is that I started this job in August of 2020, and I work in Africa, and which was like just such a great time, right, to get started as an assistant professor, and particularly as an assistant professor who focuses on international and who works in international locations. So it's only been in the last couple of years that I've really, my lab has really sort of hit the ground running. In fact, I've spent the last 18 months in Africa. So my lab is currently deep in the middle of lab work and data processing. So I'm going to have very few sort of unpublished results to talk to you about, but I'm really hoping to still raise some interesting points about that make you think about sort of how we do our work. So I'm an evolutionary biologist and an anthropological geneticist by training, meaning I sort of came to working on humans and non-human primates kind of kicking and screaming. I don't really like humans that much. But it turns out they're really interesting. So my research focuses on understanding the interplay between our genome and our epigenome, our environment and our phenotype, right? So when I say genome, what I mean is literally just the sequence of DNA in our cells, right? And the epigenome is all the regulatory mechanisms that determine which genes are on or off, when they're on, where they're on in the body, right? Are they turned on in the liver, in the brain, and how much during development, during today, tomorrow, and then how much are we doing here? So the epigenome is, we talk about it like a light dial, right? Turn it on, turn it off, with a dimmer switch to it, yeah? And when I talk about the environment, I'm talking about both the abiotic factors, things like climate, geography, and the biotic factors like animal, plant, and pathogen communities. And for humans and other primates in particular, those biotic factors also include social factors. So things like group dynamics, culture, lifestyle, right? Whether you're a hunter-gatherer, whether you're a white-collar office worker, a blue-collar worker, whether you live in a rural setting or an urban setting, all of those kinds of things. And these are the factors that sort of shape the evolutionary forces that are acting on our phenotypes, right? And on our genome, and the characteristics that sort of result from all of these interactions is our phenotype, who we are day to day. I'm going to start with sort of talking about the first project that led me into all this research, which was my dissertation, studying how genetic diversity interacts with the environment to affect fitness using ring-tailed lemurs as a model. And then I'm going to talk about the work that I've been doing in Uganda for the last 6 years, looking at how one's lifestyle and environment acts on one's genome of evolutionary time, and how changes in the environment act on one's epigenome. And then I'm going to finish with talking about another project that I have, sort of answering the same questions, but using a non-human primate species, the greater thick-tailed bush baby, as a model in South Africa. So first, right, I want to set the scene for people who I think my headphones are about to die. That's very unfortunate. I might have to switch to a set of speakers. We'll see. Let me know in the chat if you stop being able to hear me. Can we do that? Actually, you know what?
I will let you know.
Cool. I'm gonna swap them out here. We'll do one at a time. So my the first work I was doing in this arena was from 2010 to 2013, right, where next generation sequencing was just starting to become available to regular scientists, right? The cost of a human genome at that time was somewhere around $50,000. We were still multiplexing 8 samples per run. We were getting like 100 gigabytes of data per run. And to put that into context, last couple of weeks ago, I spent, I think, $20,000 to get 500 gigs of data for 132 samples. And that wasn't even that was just like a third of the samples that were on that run. So you were really limited in what you could do when it comes to sequencing. And particularly I'm working on a non-model species here. So we were really, really limited in what we were able to do, meaning that we needed to focus on specific genes of interest rather than sort of diversity across the genome. That meant that I decided to focus on a set of genes that are under pretty strong selective pressure, in particular, a set of genes called the major histocompatibility complex, or MHC. In humans, this is called the HLA, the human leukocyte antigen complex. And it's basically the immune system, right? MHC genes encode proteins that recognize the difference between self and non-self proteins, right? So they are on all of your T cells and B cells, and basically when they are presented with a viral or bacterial pathogen, or sometimes an allergen, they then go downstream and activate the immune system, right? So we expect them to be pretty diverse, and we expect that the more diverse they are, the more pathogens they should be able to recognize, meaning that diversity here should be expected to be really high, and more diversity is better than less diversity, right? In fact, we know that because of just the insane selective pressure that pathogens exert on living organisms, most vertebrates have multiple copies. Humans have somewhere between 2 and 4 copies of this gene. You can see this here, depending on which MHC gene we're talking about. Great apes have the same number. African and Asian species have even more, in particular this DR gene that's been really heavily studied. They have 6 copies of this. And South and Central American monkeys also have 2 to 3, up to 4. And in general, right, all of these genes are on one chromosome, which means that diversity because of haplotypes, right, diversity at one gene tends to correlate with diversity at these other genes because of linkage disequilibrium. But that doesn't necessarily mean that diversity here is correlated with diversity across the entire genome. The other thing to know is, so this is for humans, and this is like a chimpanzee. At each of these loci, there are many, many different alleles that can be present, to the point, and again, this is the most diverse part of the human genome. In fact, when we scan the human genome for signatures of selection, we have to exclude this section because it's so diverse. It's been under such strong selective pressure that it basically swamps everything else. It shows up as this giant peak, and nothing else shows up as significant because there's so much diversity here. Humans, it has its own website, the IMGT website, and humans have over 3,000 alleles that have been identified just at the DR complex alone. I want to say there's something like 11,000 human MHC or HLA alleles so far, and it gets updated monthly. So we're really talking about just an incredible possibility for diversity, both within an individual and across a population. And we know from a ton of research that this was a really active area of research in the late 90s and early 2000s and into the mid 2010s, that MHC diversity correlates with better health. So more MHC diversity correlates with better health, increased risk or increased likelihood of survival, and increased reproductive success, including both mate choice and offspring survival. And this includes research that's been done both in humans and in multiple primate species. So I decided I started working on this project using the ring-tailed lemur as a model, in particular because I had access to a pretty large population, over 120 captive individuals from the Duke Lemur Center, the Indianapolis Zoo, and the Cincinnati Zoo, and then samples from nearly 200 wild ring-tailed lemurs from a long-standing project happening at a place called Beza Mahafali Special Reserve in Madagascar through a collaboration with 2 primatologists, Michelle Souther and Frank Kuzo. And so what happened is we got blood samples from all of these guys. We genotyped them using next generation sequencing and basically determined that they have the same number of allele copies, or the same number of loci as humans do. They have up to 4, and it's meaning they can have up to 8 possible alleles at each location, or within an individual. Unsurprisingly, when you compare captive individual diversity to wild individual diversity. So this is the proportion of the population that has up to this number of alleles at this MHC gene. Captives in green, wilds in black, and what you can see is on average, captive individuals have approximately 2, which is unsurprising. It's a captive population that's been in captivity for at least 60, 70 years. So we expect some inbreeding, even though there is a reasonable population size in captivity in the United States. And the wild individuals on average had 1 additional allele compared to the captive individuals. And, the range was also really interesting. You can see the captive individuals at most had 5 alleles, whereas the wild individuals had up to 7. The great thing about this population, one of the reasons I also wanted to work with them, though, was that we had a lot of health and survival and reproductive data. I'm just going to show you a little bit. So we could ask about whether MHC diversity is associated with survival, in particular in a stable environment. So this is where we're getting into the environmental effects. When I say stable environment, what I mean is the captive setting, right? Captive animals are some of the best cared for non-model species on the planet, right? They receive incredible veterinary care for the most part. They are fed a much more balanced diet than the average human eats. And they are warm in the winter and kept cool in the summer, et cetera. They don't have to worry about paying bills, any of those things. So this is a Kaplan-Meier survival curve. This is the survival probability on the y-axis and the age in years on the x-axis. And a Kaplan-Meier survival curve only works when environmental conditions are stable. So first thing we start with these less diverse individuals. So individuals with 1 or 2 alleles. These are only the individuals in green. And what you see is this basically shows every time there's a drop here, that's a death. Okay. So there's no difference between, for the less diverse individuals in terms of survival probability as you age. And captive ring-tailed lemurs can live up to, I want to say the oldest one is 38. And this isn't to say that this guy didn't die. It's just that the study ended before that individual had passed away, right? But if we compare these less diverse individuals to individuals with 3 or more alleles, so everybody in green in this part of the graph, what we get is this. And again, it's not saying that these guys are never going to die. It's just that they are much more likely to make it to this age with more MHC diversity. And this isn't earth-shattering. We sort of expected this, but it was interesting nonetheless. And we also showed that it didn't have anything to do with average genomic diversity. This is average heterozygosity across the genome. It's using microsatellites, but it's still a pretty good representation. What you can see is that more diverse individuals in green are no more likely to make it to 30 than less diverse individuals in black. And again, like I said, this is a captive setting. This is a stable environment. They don't face any predators. They have unlimited water, kind of unlimited food, not always, but kind of. Vet care, et cetera. They are literally like the 1%, right? Where we're finding that genomic diversity is influencing survival when the environment is extremely stable. What about when the environment is not stable, right? What if we add in environmental instability to this picture? We can answer this question by studying individuals in Madagascar, because Madagascar is a notoriously unstable climatic environment. It is routinely hit by horrible droughts or really, really bad cyclones that cause a lot of flooding. Both of these pictures were taken in the city of Tuliar, which is about 130 kilometers from the site that I worked at, Beza Mahafali. Beza Mahafali is at this yellow star, and this is the town of Tuliar. This is all in southwest Madagascar. I want to see these 2 pictures were taken 2 years apart. This is a graph showing the rainfall at Beza, and what you'll see here is, so this is total rainfall in millimeters across the year from 1981 to 2018, I believe, or 19. And this black line in the middle is the mean, and then this is 1 standard deviation above and below, okay? And for us, these standard deviations, this standard deviation is 38 inches of rain, and this one is 25 inches of rain, which is basically comparing San Francisco to Seattle. And then when you do the extremes, so here to here, we're going from 20 inches of rain to 50 inches of rain, which is like going from Denver to Atlanta, right? In any given year. This is I'm not kidding when I say this is just an extremely variable environment where it is almost impossible to predict what rainfall will be in any given year. I have so if we look here at Beza, what we can see is there have been 3 pretty serious droughts from 1980 until a couple years ago, and 2 really extreme rainfall events caused by these cyclones. And in fact, these cyclones, what mostly happens is that the area floods and it ends up depositing a ton of silt onto all of the ground cover, and the animals have a tendency, then there's no food for them to eat. So we know that these, what we're calling extreme climate events, actually affect survival for these wild primates. In fact, this one in 1991 caused an increase in infant mortality from an average of 30 to 40% in any given year to 80% of the infants died that year. And for adult females, the average mortality is usually 5 to 10%, but that year it was 25%. So we're seeing just this doubling in infant mortality and almost a tripling of adult female mortality, which is who we are best able to follow because the males have a tendency to sort of follow females wherever they go. So I have really good survival data for this population for these years, from about 2004 to about 2015 or so. And during that period of time, we get 2 of these extreme climate events. We get a flood, a cyclone that comes through in 2005, and then a drought that lasts nearly 12 months in 2010. So the question here is asking sort of, do these extreme short-term variation in environment cause changes in the selective pressure that's acting on the genome, right? Now, the problem is the Kaplan-Meier survival curve that we used before only works on analyses you do. One of the assumptions is that the environment is stable, which clearly is not the case here, right? We should be using something called a Cox proportional hazards model, but we actually just don't have enough power to really use that model. So I'm just showing you some sort of suggestive data here, but if we graph the average diversity by year of individuals that die in black versus individuals that survive every year in green, and we compare that to the rainfall, what you can kind of see is in the year following that every year, individuals that survive on average have a higher MHC diversity than individuals that die, with the exception of 2 years. This year here and this year here. And this is the year after that cyclone and during the drought. Suggesting to me that environmental factors, right, that selective pressure on MHC diversity changes depending on the environmental conditions, such that during normal years, being more diverse is better. You're more likely to survive. But during a crappy year, it doesn't really matter. The probability of death, as we like to say, is rather stochastic. So this sort of led me to this really interesting, this is just where I started to get really interested in the impact of environmental variability on the relationship between the genome and an individual's fitness. The problem with that is that the genome doesn't change, right? The genome, your genome is your genome forever and ever and ever. It's not responsive to the environment, but the thing that is responsive to the environment is the epigenome, which is where we're headed next. So the next area of my research was looking at the environmental side of this relationship, and in particular, how the environment can affect one's human phenotypes across evolutionary timescales. So I got into looking at this one particular example of how the environment has shaped human genomes and human phenotypes, and that was studying the short stature of rainforest hunter-gatherers across the tropics. So this is a convergent evolution phenotype. It's, when I say short stature, I mean, I'm talking like everybody here. So this map is showing areas of high rainfall and heat in green, and these different populations and their heights. These are all rainforest hunter-gatherer populations. And you can see 153 centimeters, 144 centimeters, 160, 152. These are all of these individuals basically are under 5 foot. We used to call these individuals pygmies. We don't anymore. That is now become kind of a derogatory term, but you've definitely heard of them. And the population I ended up focusing on was the Batwa. They are a group of rainforest hunter-gatherers from southwest Uganda. In particular, they used to live in windy impenetrable forest in Mahinja, which is down here. They also live in the Congo, all through here, through Virunga National Park. And they have also been they also have lived in Rwanda at various points in time. And they've lived likely in the rainforest for tens of thousands of years, we're pretty sure. We don't know the exact time frame, but we do know that they split from agriculturalist populations at least 135,000 years ago, and that eastern and western rainforest hunter-gatherers split from each other at least 20,000 years ago. And what I mean when I say sort of this pygmy, this short stature, Batwa men are about 153 centimeters, which is about 5 foot tall, and Batwa women are about 145 centimeters, which is about 4 foot 9 inches tall. When I'm in person, I demonstrate this on myself because I'm 5'1". So I demonstrate that the Batwa, most of my Batwa male collaborators are about my height, and the women, like these women here, come up to approximately my chin. And I'm short. Again, I'm 5'1". So this is a pretty drastic difference. And if you compare them to this neighboring population that lives right outside the forest, the Bachiga are agriculturalists. They're part of the Bantu language expansion from West Africa, but they've been in the area for a couple thousand years, likely. And they're approximately 10 centimeters taller on average than the Batwa men and women, even though they're living literally right next to each other. These populations live within a couple kilometers of each other. And so, so they're experiencing basically the same environment, but vastly different selection pressures leading to different phenotypes. And in fact, one of the questions we always get asked is this short stature due to nutritional deficits? And the answer is no. In fact, if we do this, oh, now I'm blanking on the word. If we assess the linkage between height and Batwa ancestry across a population of about 150 individuals, what we find is that height is significantly associated with about 16 different areas of the genome, within which there are multiple genes related to growth, related to limb morphology, related to organ growth, and a couple of other phenotypes that I'm going to talk to you about in a second. And the other thing that I'm going to jump the lead here, but the other reason that we know these are not due to nutritional deficiencies is that the Batwa no longer live within the rainforest. And I can confirm, having spent about a month there actually I've spent about 6 months there in the last few years, the kids that are being born, still short, despite the fact that they're now eating basically the same diet as the neighboring Bachiga. Other areas of phenotypic divergence that are within some of these areas under selection include limb morphogenesis and development, right? Growth factor binding, organ growth, but also genes associated with cardiac health and function, in particular, regulation of blood pressure and cardiac muscle tissue development. I can talk about why we think that is present at the end if anyone has any questions, but also immune system function and development, which we know based on some experiments that the Batwa, their immune system response, their immune functional response to viral pathogens and bacterial pathogens is a little bit different from the Bachiga that they live next to. And I'm giving away I sort of alluded to this before, but the Batwa have lived in windy impenetrable forest for tens of thousands of years. They have what we are now calling a rainforest hunter-gatherer genome. They used to live a rainforest hunter-gatherer lifestyle. This is what I consider a genome environment match. But in 1991-92, windy impenetrable forest was created as a way to preserve the endangered mountain gorillas, and the Batwa were forcibly evicted at that point in time. They were resettled outside of the forest in little settlements interspersed between their Bachiga neighbors. And they were forced to adopt an agriculturalist lifestyle. In fact, to the point where they're not allowed in the forest anymore. It is well, ask me about it later if you're curious. Um, so we now have this situation where the rainforest hunter-gatherer genome is living an agriculturalist lifestyle, putting them in this genome environment mismatch. And this mismatch is just extensive, right? This is what windy impenetrable forest looks like. This is what the Batwa, this is how the Batwa used to live. This is a woman named Flora Baraku. She has no shoes on. She is using a staff every day. This is a picture of her every day as she goes in to a patch of forest that's no longer in Bwindi and gathers wood for a fire that she's going to cook over. And here you can see where the Batwa live now. They're living on fields with banana and plantain and manioc. And they're sending their kids to school. This is the school. The school's over here, actually. This is the community meeting house, and this is a health clinic. They're receiving health care. They're using antibiotics. And so this has really changed absolutely everything from diet to sun exposure to activity level, disease exposure, all of these things, right? Imagine, all of us know or either have kids who they've sent to daycare for the first time or have friends who've done it, right? And remember how sick you are like the first 2 years your kids are in daycare? Imagine if you're part of a population that has never spent a significant amount of time in enclosed spaces with other people, right? Their kids, there was a period where their kids and they were basically sick all of the time. Um, and I hate to use this term, but this offers kind of a natural experiment for studying how environmental changes, how an individual might acclimatize to these environmental changes. And so this is sort of what I'm looking at, right? And we're focusing on the epigenome because the epigenome is the thing that is responsive to the environmental context, right? The epigenome is controlling transcription, it's controlling gene expression, and it serves as a bridge between an individual's lived experience and their genome to generate phenotypic variation, right? And so this is we're studying the Batwa now uh, after having lived in an agriculturalist lifestyle for approximately 35 years. And interestingly, we have this really interesting generational divide where individuals who are over 45 actually were raised in the rainforest. They did most of their development up to 13, 14, 15 in a genome environment match. They lived in the rainforest and then they were evicted as adults. And then we also have this generation of younger Batwa, individuals under the age of, say, 34, 35, who have spent their entire development and entire adulthood in this agriculturalist environment, but they have the same rainforest hunter-gatherer genome as their elders do. And then we also have this comparison population of the Bachiga, right? This agricultural population that have spent their entire development and adulthood in a genome environment match. And so this is the situation in which I'm currently working. The project goals, this project has 3 goals. And the first one, and actually this one was always kind of in the ethos of this project, but it became really formalized over the course of the pandemic when I was spending a lot of time talking to my collaborators in Uganda without actually, being able to do the work. And so this then crystallized into one of the formal project goals, which was engaging the Vato community in the research process from planning all the way to interpretation. The second goal was to quantify epigenomic differences across age and lifestyle between the Batwa and the Bachiga. And then the third project goal was one that was suggested by one of the collaborators. And that was part of of fulfilling project goal 1, which is to contextualize the lived experience of this transition via recording oral histories of Batwa elders and youngsters, which we are actually in the process of doing right now. So this, the way we've approached the collaboration, we have 3 collaborating nonprofits in Uganda. First, Bwindi Community Hospital, which is a nonprofit, pretty large hospital in the district right outside of Bwindi. And then two nonprofits, the Batwa Development Program and the Action for Batwa Empowerment Group, which are leading the on-the-ground efforts in Uganda. And in fact, actually, the CEO of ABEG, this Action for Batwa Empowerment Group, came to UC and gave a talk in 2023, and she's actually going to be giving a virtual talk here in Cincinnati in a couple of weeks, which I'll provide more information about at the end. But we hold pretty monthly Zoom meetings with these collaborators to just check in with everyone, make sure that we're on target, make sure that there haven't been any issues, and make sure that the research participants are hearing about the progress. Because after we meet, research assistants from ABEG go out and keep the community up to date. And what do I mean by that? So I spent over a month and a half just doing community engagement discussions in Uganda. No research, just, hey, we're coming to your settlement. We want to talk to you about this project. We want to hear what your thoughts are. We want to hear what you're worried about. We want to hear what you're excited about. And then we did the same thing prior to every sampling site, prior to any of the individual consent discussions. We spent about an hour and a half to 2 hours discussing the project, the possible results, and our commitment to returning the results and getting their approval on the interpretation before we publish anything. Our core team is 90% Ugandan. This is from 2025. This is the research team. There's me, and then this is Sylvia. This is the CEO of ABEG. Here's a couple of our nurses who were collecting samples, and the rest of these are research assistants. And this is really important because Sylvia, and in particular, so this is Sylvia, this is Edward, this is Honest. They are all Mutual individuals. They're all part of the Batwa community. And this is Haven and his wife Diane, and Davis and Stephen. So these three here, they are Batwa. These four are Bachiga. And that's a really critical part of this project, right? Is that by having co-PIs and research assistants who are community members, the community feels comfortable elevating their concerns, and asking any question that they're worried about. I got a lot of questions that we wouldn't necessarily expect here in the US, but things like, are you going to sell my blood? Things like, are you going to put a curse on me? Things like, are you going to test me for medical conditions, even if I don't want to be tested? And having community members as co-PIs really helped to alleviate a lot of the concerns that some of the community members had. And then, okay, so that was, what we're doing with Project Goal 1. When we get to Project Goal 2, last spring, I spent 3 months there and collected 400 samples from 400 participants, 200 Batwa and 200 Bachiga. And the cool thing about this is that in addition to just generating gene expression and methylation patterns, one of the things that I'm really committed to is helping to improve representation in research in general, both of participants and of researchers, right? So if we look at the individuals that have been involved in genomics research, they're overwhelmingly of European descent, right? Followed by East Asian. And then when we get to African, Hispanic, any of these other groups, right? Their representation is so small that it is less than 3% of research participants, despite the fact that the global population, they represent a much, much larger proportion, right? And when it comes to researchers, this is looking at the number of genomics papers with authors from the United States or African authors. What you can see is that until very recently, there were virtually no African authors publishing in the human genomics research space. This is being in part addressed by projects like H3 Bionet, H3 Africa, and Malaria Gen. But I'm hoping that my research will also help to contribute to the increased representation of diverse participants in this kind of research. And then finally, again, this is Sylvia Kokanda. She's one of the co-PIs, and she's the CEO of ABEG. This is her daughter, Kayla. This is us collecting samples and consent. Kayla makes an excellent desk from which to sign consent forms. Um, the and she was the one who really brought to me back in about 2022 this very last project goal. She came to me and one of the questions I had been asking her was, what do you guys want out of this project besides the research outcomes, besides being hired as research assistants and training and getting and having money funneled into this community? What else do you all want? And what she said was, a lot of our elders, we're losing them. And we're losing their knowledge and their experiences and their stories of living in the forest. And so we came up with this idea that we were going to start recording oral histories of these elders, and that we were going to preserve their experiences. And so I recruited two collaborators, Dr. Daniel Murphy and Dr. Kate Nicole Hoffman. Dan Murphy is an anthropologist at UC, and Kate Nicole Hoffman is a postdoc at Pew's, the Center for Public Engagement with Science. And Dan Murphy explores the relationships between humans and their environments, focusing on the cultural, political, and economic dimensions. And Kate Nicole Hoffman studies how humans conceptualize what nature is and how they experience nature. So they were really sort of the perfect people to bring onto this project. We spent about a month in this May recording. I don't know if this is going to play or not. You guys will have to let me know if you can hear it. Can you hear this?
The video is playing, but I don't have any sound.
Oh, there's no sound. Okay. Oh, bummer. Okay. Well, so she's talking about how this is Adrienne. She's one of the Batwa elders. She's talking about how, sorry, I have a visitor who is furry in my office and he's decided to start barking. But she's talking about how the Batwa elders weave native grasses to make crafts like this platter here, and what the process is for dyeing the different colors, how she goes ahead. I can actually hear. Yeah, we can just sort of play it. She's so how she's weaving through this thing, through this for this platter, and working to sell these crafts through the cultural center that ABEG is working to develop. She's using a needle here, but they used to use just a sharper and thinner grass stem. Some of these you can see are the yellow she's got there is a pretty unnatural dye color. But the white, and then in this one that I'm holding here, the brown and the green are all natural, are from natural plant fibers. And so over the course of this period of time, we recorded over 100 stories from over 40 participants. And this work is continuing throughout the course of this next year without me, Dan, or Kate. In the last few minutes, I'm gonna turn my attention to the last project, which is using another non-human primate model, the greater thick-tailed bush baby, to look at short-term environmental change, right? So the Batwa, the goal of that project is long-term environmental change, sort of single permanent environmental change. I'm using the bush babies to look at environmental variability over space and over time. I wanted to go back to Madagascar, but unfortunately, Madagascar had its third coup in, 2 decades. And we ultimately decided that it was just a little bit too unstable to work there. So instead, I now work in South Africa on this species. This is Autolima crassicaudatus, a primate relative to humans, and they're pretty widespread. So this is their species range, all the way up to Uganda, very southern tip of Uganda, and all the way down to the very top of South Africa. They're primarily forest-dwelling species, but they are pretty agnostic about the type of forest. They'll take tropical, they'll take subtropical, they'll take montane, anything. And you can also find them in tree patches on savannas and in agricultural land, things like livestock farms. They're the largest bush baby species, somewhere weighing anywhere between 1 and 2 kilograms, which is 2 to 4 pounds. They eat primarily insects, but also tree gum, and then occasionally fruits and seeds. And they're nocturnal. They're considered solitary, although we have quite a lot of evidence of them sleeping together, cuddling, feeding together, etc. So they're way less solitary than we think they are. They reproduce annually, giving birth to 2 to 3 offspring per year. In captivity, we know they live up to 22 years, but in the wild, we've actually documented individuals living up to at least 12 years old. So the data from this project is coming from a long-term research project at Lajuma Research Center this is South Africa. This is the Limpopo province up here. This is northeast South Africa. And this is the Saupansberg mountain range. Lajuma is located right here. This is one of the bush babies we caught 2 years ago. And this project has been ongoing since 2013. This project has captured nearly 300, and actually I think it's over 350 at this point, over 350 captures of nearly 200 unique individuals with a pretty good mix of males and females. Most of them are adults, but we do catch a few subadults every year. And the exciting thing is that we've actually captured individuals who've transitioned from subadult to adult. This is the number of individuals we've caught every year. So starting in 2013 up through this year, with new individuals in blue and then individuals we had previously caught in black here. And you can see that we get a pretty good mix of new and sort of repeat offenders. On average, we capture about between 20 and 40 individuals, depending on how many nights we're trapping. We have a number of individuals that have been captured up to 8 times. We've got 30, 40 individuals that have been captured at least twice, and 10 or 15 individuals that have been captured up to 8 times. And you can see here, this is a number of individuals with years between captures. And we use this as an assessment of age. The youngest we ever capture them is about 8 months old. They're still subadults. And by that, we can assume that if we captured you, if there were 12 years between the first year of capture and the last year of capture, we can assume that you are at least 13 years old because you were at least 8 months when we caught you the first time. And so this is where that age estimate of individuals in the wild comes from. Now, that's all the species, but Lajuma itself has a really interesting range of annual variation, in particular in rainfall, similar to Madagascar, in that it's not quite as expansive, right? You can see these numbers the Madagascar numbers went up to 1,200 and down below 400. Lajuma doesn't get quite that amount of variation, but you can see that there is pretty decent variation here. Between very, very wet years at nearly 1,000 millimeters down to about 400 millimeters. And the thing to notice here is because we've been trapping these individuals for so long, we have weight data for individuals year to year. Female adults are in purple, male adults are in black. They're black squares, females are purple circles. And this is trapping them at the same time of year every year, so we know there's seasonal variation in weight and in rainfall. But the thing to notice here is these yellow arrows are pointing at dry years and the green arrows are really wet years. And what you see here is that these yellow arrows dry years correspond with nearly a 10% decrease in weight. So a 50% drop in rainfall corresponds to a 10% decrease in weight. Or correspondingly, a 400 millimeter increase in rainfall corresponds to a 10% increase in weight on any given year, which is a really important indicator that obviously environmental variation is having an effect on phenotype. So the thing that I'm interested here is looking at the effects of age and sex on things like gene expression, on methylation patterns, and then also the effects of environmental condition, both currently and also the effects of environmental condition at birth. Because we know from things like the developmental origins of disease hypothesis that the conditions at birth are expected to have an outsized impact on physiology, phenotype, health, et cetera. And we might get to see those things biologically encoded via the epigenome. So far, our goal is to capture individuals every year for several years, including catching individuals from our year one cohort during year two, during year three, et cetera. And we've actually managed to do that. And the idea is to do both a cross-sectional and then a longitudinal study looking at both response to environmental variation and the plasticity of this response, right? And we can do that because of the number of individuals that we get repeat captures. And then I want to correlate all of those things. I'm trying to speed through here because I know that we're about out of time. We want to correlate all of those things to physiology and health, that each of the animals, when they're captured, gets a full physical exam, and samples are taken. We measure things like body fat percentage, muscle tone, tooth decay, parasite level, et cetera. And then also we look at things like whether they are seem to be reproducing or not. So far, so 2025, we caught 48 individuals, and this year we caught 38 individuals. We had a slightly shorter capture season this year. But we ended up with, those numbers are close. They're not quite accurate, but they're close. 99 captures of 80 different individuals, about 20 of which this year we caught that were previous captures from the year before. All of those samples are currently in the process of being sequenced, and we're really hoping to have data within just a month or so. And like I said before, for the human work, I actually received, the download of 100 or 500 gigs of data from RNA-seq data from about a third of the individuals that we sampled, and we're in the process of sending off the rest of them. And I received those data about a week and a half ago, so I don't have any results of any of this to show you. But I hope this gives you sort of an overview of why I'm doing this kind of work and where we're hoping to go from here. And with that, I'm going to take a second to thank the Batwa and the Bachika communities and the Bush Baby Capture team. This was one horrific night this season when we caught 10 animals and processed 10 animals in one night, which we will never do again, because that was a 14-hour day, and never, ever, ever again. And with that, I want to thank my collaborators and my funders and the research centers that I work at. And then I want to highlight something that's happening locally related to some of these projects. So the Clifton Cultural Arts Center is currently running an exhibit on Batwa ancestral weaving, so story and craft. The exhibit is open. The CCAC is free for individuals to look at. You can see some of the exhibit here. And we'll actually be holding a panel discussion with Sylvia and some of the Batwa elders on August 8th, which is a Saturday at 10 a.m. at CCAC, which is just up above north of Burnett Woods. So it's very, very close. And with that, I am happy to answer any questions that anybody has.
Thank you, Dr. Grogan. That was a great talk, and I'm excited to see how your work continues to grow. Can you hear me okay? This is Kelly Brents. If you can.
Yeah, Kelly, you're a little crackly, but I can hear you.
Okay. What, just for some of the students on the call, like, what do you think has been one of the biggest learning moments for you as you do some of this international work? I know it's kind of away from the science, but like just kind of reflecting on this.
Honestly, I'm so glad you asked that question. One of the biggest learning moments. One of the biggest learning moments for me has been that we as researchers often do not spend enough time in the communities of that we are researching. We don't, and I think that probably does not apply to this audience, which I find really interesting. I'm used to talking to genomicists, like human geneticists and evolutionary biologists who maybe work on humans, but it the most important thing that I learned sort of was that if you that the communities you're studying are the people who know themselves best, which I recognize is an incredibly obvious conclusion, right? But understanding that they are the best people to help you interpret the results, and that you're doing science and them and yourself a disservice by not involving them from the start in the idea generation, in the data collection, and in the interpretation. And even if that means that the research goes slower, it will be richer and more ethically grounded than if you sort of followed the traditional outside objective observer model that many of us more senior researchers were trained on.
Yeah, I mean, I would agree. I would say just, reflecting back, like some of my own hesitations, because I do some epigenetic work myself, is like, do we think our participants and the folks that we're working with understand these concepts, right? Or is it my job to make sure they understand these, what we would call complex, concepts or ideas? And I'd love to hear your thoughts, since I don't see any other questions or hands raised, like how you kind of go about that conversation with community partners or participants and things like that.
Yeah, so I don't worry so much about making sure that they understand what a methyl group is, right? What the genome is, what the epigenome is. Like we don't talk about, in fact, actually I don't talk about genetics. And I will say my situation is a bit of an extreme one. I'm talking to a population of which about 40% of them are still illiterate. Of the remaining 60%, about 30% can basically just print their names. Very, very like they're very, very few of these individuals have made it through high school. And so you can't really, DNA, RNA, like no, that would take so long to explain. So instead what we talk about is our bodies, right? Because that's where the results are manifesting. They're manifesting in your body, in your health. And so we talk more about that than we do about the specific. Does that make sense, Kelly?
Yeah, absolutely. Yeah. I mean, I think that just underscores why it's important to understand, right, your audience and sort of what they would gain to